It is the college pleasure and the department pleasure and my personal really pleasure to make this introduction. The best part of my job really, and actually the most difficult part, is to find the right people and provide them with a platform really to flourish and achieve their aspirations. So it's wonderful for me to be in this situation. Give me the honor to introduce you. And before saying any housekeeping, I think having Rakesh here is actually not just something wonderful to have, but using as a model for other subspecialty to say we are not one-man band in each specialty, but we can have multiple PI, multiple professors in one specialty with a very defined roles and a space to go for the next stage, and they do things to change career and to change the field. So we're using urology as a model, not just, and your work as a model of what we do. And the other thing is, I don't think we can start anything without really to say thank you for the family, because those are the people who really bear the burden of all what is happening. Parents, which they will be very proud of you today, and the family similarly will be proud of the husband and the dad. So I think this is really, I really means a lot to you and means a lot for us too to have you here. So I think you will hear from Rakesh, from the king himself, what it's all about. So I'm not going to say much about Rakesh, later than you will see what he has done and what he achieved and what he will achieve at Imperial. I have to say a few housekeeping points. And actually, I need to thank you, the one who brought and mentored you to be here, and Naeem, I'm sure you will hear about from you. But I think it is really wonderful to have you here, to see your handwork now flourishing and becoming your mentorship achieving more. So there is emergency fire briefing. So in the event of fire, you need to follow the exits, and as far as we know, there is no test fire alarm. So if anything, it's real, not a test. And secondly, the event will be recorded, and there will be online questions. So, and I think if you are going to chair the few minutes of Q&A, but if you have any question, you let me know, and then we will find things. So before hand to Rakesh, special thanks to the whole urology community at Imperial, clinical and academic, and Hash and other people, to have this environment to allow us to have Rakesh here. So we're looking forward to hear from you. Okay.
George, thank you for your generous words. They've been really appreciated. Hashim Ahmed, sat next to George, Professor Ahmed here, approached me getting on to, I think 4 years ago, about coming to join his endeavour here at Imperial, and introduced me to George Hanna. And I have to say, I was so impressed with the clear ambition that George had to take what was already an outstanding department of surgery to the next level, to become a world leader in surgical research, and I think it's well on that way. And when I got to join Imperial Urology, I can't tell you how thrilled I was to join 4 professors of urology, to become 4th professor of urology, working with Hash Ahmed. We've got Marcus Drake here, and we've also got Suxminas covering the breadth of the main cancers in urology: kidney, bladder, prostate, and also benign urology, covering functional urology and andrology. I think I can't stress enough how proud I am to be part of an absolute powerhouse in the UK for urology research. With that, I'm taking this opportunity to talk about something that is central to the scientific method. What we do all the time is challenge ourselves, challenge what we think is true, challenge dogma. We bring a critical eye to what we think we know. And today, what I'm going to do is share my story about how I've done that through my career, encourage you guys to do the same, and to present that story through the lens of my prostate cancer research. Now, prostate cancer is very much in the public psyche, primarily in and around high-profile cases, and perhaps the most recent case of prostate cancer that we know of is Chris Hoy, an Olympian, a young man, and sadly diagnosed with advanced metastatic disease. And like many, it begs the question that I experienced when I was a junior doctor, looking at patients in my clinic. Why do patients or people, men, get prostate cancer? And why do some men present late? We know that if you present early with prostate cancer, it's imminently curable. Let's have a closer look at that. So if we were to look at the demographics of the UK, we know that for cancer deaths, prostate cancer accounts for the second highest causes of death. We know that of the 50-odd thousand men that present every year with prostate cancer, approximately 40% will have advanced disease. Half of those with advanced disease have metastatic disease. Sadly, they're going to die from prostate cancer. So why do some present early and others present late? Here, this schema hopefully talks to why that happens. We've got time here, and what we've got is growth of prostate cancer over time. And we see some cancers grow very slowly in this curve, and they're microscopic. In fact, if you live long enough, it's very likely that as a male, you'll end up having prostate cancer. It's just that you'll never know about it. Then there's others that grow more rapidly, and they lead to disease that presents. It's localized, it's curable. What we want to capture are these men, which are currently being missed. They develop prostate cancer, but the growth is such an explosive growth, this exponential growth, meaning that they go very quickly from microscopic disease to metastatic disease, often before symptoms manifest or presented with symptoms of metastatic disease. We need to do better for these men. And to do better, I think trying to catch them in this space is difficult. We need to understand a little bit more about what's happening here at that histological, that microscopic level. We need to understand what the molecular mechanisms are that change these cells from normal cells into cancer cells. We need to understand those cells that are vulnerable to malignant transformation. So out of those millions of cells, hundreds of millions of cells in individual organs, which ones are the ones that transform? We think those cells are stem cells. So let me talk you through the organization of stem cells in the adult setting. So in an organ such as the prostate, sitting just underneath the bladder here, we have a distinct pool of stem cells. They work as a factory to generate daughter cells that differentiate, and these differentiated cells are the ones that make up the tissue of the organ, and these do the work, they turn over. There's a finite lifespan, and they're wasted, they're turned over, and they're lost. And as they're lost, the stem cell has a role of replenishing that tissue. So there's a balance between turnover and replenishment. Stem cells then proliferate throughout the life of the tissue, and that means they are the ones most likely to accumulate those molecular changes, those malignant malformations in the genome that lead to malignant behavior. So that curiosity very early on in the 2000s set me off on my PhD. I was chuffed to be awarded an MRC fellowship, and I did this very exciting body of work in a space that was relatively new at the time, in and around stem cell biology and cancer stem cells. We have our supervisors, Professor Craig Robson and Professor Hing Lee Young, and Craig and Hing were brilliant supervisors, but also great at asking the question, how true, how valid is this? And they would always ask this painful question: how confident am I that I'm looking at stem cells? They were actually right. I based my definition of stem cells on how cells were behaving in a plate, cells that turn over quickly for a long period of time. In fact, that's not a good enough definition for stem cells. And with that, I realized that as I was setting off on this journey, a career in academic science with prostate cancer at the very heart of that endeavor, that I hadn't quite got the ground truth, and I needed to work out what really is right and wrong. As the wise man says, you build your house upon the rock. So I looked across at the archetypal model back then in the mid-2000s, late 2000s, and that was the epithelial model for the colonic crypts. We have these crypts that then give rise to these villi, which is where we absorb all our nutrients in the gut. And we think the stem cells reside at the bottom here, proliferate and populate the cells that subsequently are extruded at the top. So there's turnover occurring. The stem cells are the ones that persist. They can accumulate mutations. Although this model's very well described in the intestine, it's not so well described in the prostate, and you've already heard that a lot of that was based on this phenomena of doing studies in labs in Petri dishes. One of the things we've started to appreciate is that if you take cells out of their normal environment, then they behave differently. The gold standard was being described around this time. The gold standard was to undertake in situ characterizations of stem cell biology, looking at determining the fate of those cells, showing that multipotency and that ability to be sustained throughout life. And this is work done by Cleavers, and this seminal work showed that in genetically engineered mice, you can take markers for the stem cell in the colonic crypt, LGR5, mark it with a blue dye, and over time, that blue dye is transmitted to daughter cells, then migrate and populate the rest of the crypt. This is a definition of stem cells. This is how we should be doing it. However, there was a problem. The problem was we can't genetically engineer humans, and so we can't put these marks in to do the study. In the mouse, the prostate is significantly different to what we have in the human setting. So although we can learn quite a lot from the mouse models, they're not a great model to translate back into the human setting. However, a little bit of luck. When I was in Newcastle, just across the road from the cancer centre, we had a world-leading mitochondrial biology lab headed by Sir Doug Turnbull. And mitochondria are the batteries in your cells. They make the energy that we need for cellular functions. And the mitochondria that are in these are distinct from the genomic central DNA that we're used to. These mitochondrial DNA sit separately. They're not as complex as nuclear DNA, and these are much more vulnerable to mutation. And you can detect those mutations using enzyme histochemistry, because the enzymes that they code for don't work so well. So similar to what we saw a moment ago with the blue marking stem cell fate, in human crypts here, we can see that using the enzyme histochemistry stain, that you end up having a blue ribbon running from the crypt, running along, indicating that these are clonally related, derived from the stem cell at the bottom here. Fantastic. We now have a model to look at the human prostate and to define its stem cell biology. And this is where being a surgeon scientist is an advantage, because we're at the cutting edge of surgeries, and with the generous consent of patients, we can access tissue and we can undertake these kinds of molecular characterizations. So off I set to do this big body of work, and I massively underestimated how complex the human prostate is. This is a prostate that I removed during a cystectomy. That's removing someone's bladder for bladder cancer in a male. The prostate comes with it. He generously donated that for an MRI study. And those are individual ducts and glands running all the way through. This was going to be a nightmare to handle. And it was almost an overwhelming burden of work. But we had, again, another bit of luck. What we realized, looking back at some of the historic studies of histology in the prostate from McNeil, that the prostate is actually comprised of individual glandular subunits. It's a compound gland. So it's a bit like a road with trees lining along that drainage point, the road, the urethra in the middle. So we can look at these as individual glandular subunits, similar to those individual colonic crypts I was showing you earlier. So that allowed us to be able to undertake those reconstructions. It was like searching for a needle in a haystack. We were staining whole prostate sections, and this is just partly what you can see. And you find these blue dots, and then we spent ages tracing them section by section by section by section. These big glands are sectioned at 10 microns. There can be thousands of sections to go through a whole gland. And then we were painstakingly reconstructing this. The software available wasn't able to perform this. This was part of my clinician scientist fellowship awarded by CRU UK and the Royal College of Surgeons. Rather proud to say I was only the second surgeon ever in the country at the time to be awarded one of these prestigious fellowships. And we found these long-ranging clones that run all the way through the branching structure from here, near the urethra, all the way into the branches. So having done that work, we ended up making a number of powerful discoveries. I want to give a quick shout out to the people that did the hard work here. We've got Laura Wilson, our technician, and Mohammed Mode. And these two were a complete powerhouse, actually, in terms of generating this work, generating this data. And at the time, there was also a hugely exciting and an energetic collaboration forged with Ben Simons at Cambridge and his super bright PhD student, Eduardo. And what I need to thank Simon for is a certain approach to scientific inquiry, which remains with me today, about how I approach science and scientific questions. So what did we find? We found some remarkable findings. We found that in the branching tree, we have these clonal marks that start near the urethra where we think the stem cells sat, and they make a clonal pattern that runs all the way down into the very periphery of the prostate. We also identified where these started in the basal cells, and we think those were the cells of origin. We also looked at the biophysics, the spatial arrangements of these glandular structures, and working with other colleagues that were experts in breast and kidney branching, we were able to come together with a mathematical framework for biological branching in development. So some really exciting work that came out of that. During review, there were some really interesting questions that were being posed about this work. This was in review for getting on to 18 months. It took a long time for us to get this out because there was criticism around these clonal marks. The question was, are these clonal marks being acquired with age and therefore marking adult stem cells? Or were these clonal marks potentially acquired in embryogenesis and we've only spotted them late on in life, but they're embryonic marks? I remember defending it quite hard. They can't be. But there was a niggling doubt, and this is where you have to be honest with yourself. It's better to go back and be leading on some of the uncertainties in your work than other people catching you out, really. And I think I was honest enough to have a look at that. And there was a challenge. We didn't really have a way of defining marks later on in life, and this is all we had. But around that time, I was part of this consortia, this really exciting body of work looking at the mutational landscape across multiple organs in the human body, led by Raheli Raberi and Louisa Moore from the Sango, along with Mike Stratton and Peter Campbell. And what they were doing was identifying specific mutational signatures, combinations of mutations that were related to age These were clock-like mutations, so they occur with increasing frequency with time. And we looked at our prostates. We looked at a number of prostates, and over age, we saw a similar linear relationship. These are clock-like mutations that can be used to infer dates, epochs of certain biological processes. Special reference to this patient sample here, a 59-year-old, where we did a super deep dive, over 400 whole genome sequences for multiple laser capture areas. And when we look at how these mutations relate to distance from the urethra to the periphery, we see there again there's a correlation with the mutational burden. And to corroborate that, we also looked at telomere length. Telomeres at the end of the chromosomes, they shorten with cell division, and we saw that those inversely correlated with the mutation burden, implying that we do have a spatial temporal relationship in the tissue. So really, really powerful stuff. We now have a way where we have dynamic marking of mutations occurring, and it gave us an opportunity to go back and look at patterns of mutations that could be related to certain time points in an individual's age. So we started this work here where we got whole prostates just cleaved in the middle here to fit on the slide, complex reconstructions, laser capture microdissections. We track along these ductal trees. We undertook genomic profiling, clustering together mutations, and then we were providing those that information to then sit and overlay onto histology. This was part of another, a fantastic, brilliant collaboration that we had with Peter Campbell here at Sanger and another very bright PhD student, Sebastian Grossman. And Sebastian took those 400 laser capture whole genome sequences, and as we look at them on the map here, it's a heat map. It's a bit complicated, but all I want to point out is that on the x-axis here we've got all the different laser captures, and then we've got clusters of mutations shown on the y-axis, and we can see there's clustering occurring. These show that there are shared mutations across spatial distributions depending on where the laser capture site was, and we also saw nested mutations occurring. For example, there's a cluster running along here that's shared amongst all those, but down here you can see there's another subcluster that's shared amongst a smaller number of samples, and then another one here, and so on. What we can do is represent that data, and we do that here, looking at here an x-axis, looking at mutational burden. What we've got is a particular clade of mutations, which I'll present again here. And these mutations are occurring very early on in life, in embryogenesis. The subgroup within that has mutations that occurred that time around puberty, and then there's those that occur in adulthood. So we can take these epochs in time and look to place them back on to those histological structures. And what we saw is that we can get various levels of these mutations being expressed throughout the prostate, and not surprisingly, those that date to embryonic mutations, they are distributed throughout the prostate acinar structure. Those that relate to puberty are much more spatially restricted, and in fact they were side branching parts of the anatomy. That's completely in keeping with what happens in puberty for young boys, adolescent boys. As they go through puberty, there's a spike in testosterone, there's a surge then in proliferation, and the prostate gland that's relatively quiescent since birth has a proliferative burst, and we see this extra gland formation. During adulthood, we see much more restricted clonal patches as the stem cell activity acts over a smaller domain. So we've learned about branching, we've learned about geographical restriction of stem cell potential with aging. In the list of mutations, we did find a FOXA1 mutation, a canonical driver of prostate cancer. This is a mutation that's absolutely fascinating because we know it's a bad one, and when we looked at when it was accumulated in this man, his gland is histologically normal, but he does have a driver mutation. It was something that occurred in puberty. It's remarkable to think that the fate of prostates, risk of prostate cancer could be fixed as early as puberty. So we now have a revised stem cell model. We can now describe embryonic development, where we get cord-like structures that grow into the embryonic stroma to make rudimentary glands. During puberty, we have side branching occurring, driven by focal stem cells. And at this point, we might see driving mutations fixing. During adulthood, later on in life, what we have is much more restricted areas of clonal maintenance by the stem cell. And you may have a driver event which has some selective advantages that takes up a larger area. We've now described a baseline. We have a true ground foundation for us to build on. And in the next level, what we want to do is start exploring what happens in cancer. So what we've got is this arrangement where there's a spatio-temporal relationship within the histology of the gland. On the end here, we tend to get cancers in the periphery by looking at the mutational burden in the periphery in cancers and the adjacent normal structure. We should be able to, a bit like archaeologists, be able to dig further down, and as we move further and further away, we should be able to go back in time and identify mutations that are pre-malignant changes, and then those that are their transforming events. The idea is to look at whole prostates. There's a video here that unfortunately doesn't work at the moment, but it's an MRI scanning through the prostate, illustrating the branching ductal anatomy and how we can identify those areas of cancer for us to laser capture and to do these studies. That's looking forward now. We have this fantastic program of work. It's called EPI-EVO. This will be running for the next few years. It's a project I wrote primarily with Charlie Massey from Cambridge, and sadly Charlie's no longer with us. This project's affectionately called Charlie's Project. Harvey Debs stepped in from Cambridge and will be ably leading that part of the biology, and I'm excited to have a renewing of that relationship with Raheli to work on this project. Also want to give a special shout out to my team here in Newcastle, Craig Robson and Luke Gorn, who, although I've moved from Newcastle Imperial, I remain an honorary chair there, and that's a very vibrant, rich relationship that will carry on at the heart of this project. So I've been describing rather static models where we infer quite a lot by looking in those tissues and then trying to go back in time. But what we like as scientists, as biologists, are systems, models where you can have a relationship between A and B, and you look to perturb that relationship to show that there is a strong connection there. We like to have cell lines, we like to have tissue, viable tissue to work with. And that was something that I had an eye on back in the late 2010s, and we undertook this really exciting bit of work. My group became the first group to take prostate tissue and reprogram it into an embryonic-like state, an induced pluripotent stem cell. And you might ask, why do that? But the induced pluripotent cell is ultra-proliferative, it's easy to manage, easy to manipulate, and you can do quite a lot of science with that. And then what we showed is, with this work, is that we can differentiate those cells back into the prostate, meaning that we can turn on prostate cells, prostate tissue, like a tap when needed. Thanks here to Dipali, who did most of that work as a PhD, taken over the line by Mohammed in the end, and then Emma Curry here did most of this work as a PhD, again taken over the line by one of the postdocs, Natasha. If you look at the tissue that we can generate from that approach, this looks indistinguishable from what you see in the histology from human samples. It's a really faithful model to generate prostates. It was an opportunity to work with Simon Hayward at Vanderbilt, and now at Chicago, a leader, like a godfather in prostate developmental biology, and that was a very exciting collaboration. And this work helped Natasha go on to get her fellowship from PCF, a really prestigious award, as she built her career. So what have we been up to more recently, having developed that model? Well, what we've been doing is looking at genotyping prostate cancers, defining the molecular makeup of those really aggressive cancers on those exponential growths that tend to present with metastatic disease. And having done that, we can do this theoretical on an individual patient level, we can then go back to our iPSCs, undertake gene editing with CRISPR technology, and we can make a designer prostate cancer based on an individual's drivers, undertake lots of characterizations and help develop new approaches for diagnosis, for treatment. That work is being led by Adriana Buskin, who's one of our early career researchers in Newcastle. She's an amazing rising star in the space, a previous 3 Rs fellowship holder and more recently a PC UK fellowship holder. This work's been funded by the John Black Charitable Foundation and the Prostate Cancer Foundation. So, busy slide here, not looking to explain the detail on that, more to point out we've got lots of organoids that we can grow within the lab now. They form tissue-like structures, so they're not just simple cells on a plate. We've looked at important drivers, so P10, P53, MYC amplification, AR amplification, and we have different combinations of that making up a large fraction of those genetics that drive aggressive cancer. And when we compare them on a single cell level with cancers taken from direct patient biopsies, we know that this faithfully recreates that biology. So really exciting, powerful tool that over the next few years we'll see come into its fore as it starts delivering new insights into prostate. So you've seen me here as a preclinical scientist, and of course I'm a clinician scientist, and I do have one foot in the clinical space as well, and it's given me a really exciting opportunity to be involved with clinical trials. When I first started as a consultant urologist, I was doing a lot of bladder cancer work, and a bit of a pivot here, I appreciate, from prostate, but I just want to take you through another example of challenging dogma, challenging what people are presenting to be factual or strong evidence. We were looking into the bladder here. We have this cystoscopic view, and of course I think we make out there's a bladder cancer even to the untrained eyes. Bladder cancer is highly recurrent. As we resect this away, they tend to recur. Why does that happen? Well, we think it's incomplete resection. Surgeons think they're better than they are. It's a big problem for surgeons. And 20 to 40% of recurrences may, in fact, be related to incomplete resection. The idea was if we could see better, then we would do a better job of removing those tumours. And one of the technologies that were being posited as being really powerful in this space and started to break into clinical guidelines, international guidelines, was PDD, photodynamic diagnosis. And what we do is put a fluorescent dye into the bladder. It's taken up preferentially by cancer cells. And you turn on a fluorescent light, and look, this is what happens now. So you turn on blue light, and things glow pink red. There's the tumour, and many of us would have completely missed the flat tumour here. This is a very commonly used picture, shows how powerful this technology can be. But around that time, we were undertaking systematic reviews, and a lot of people were showing in their systematic reviews these kind of technologies, either PDD or other augmented visual technologies here with Rajan Viratapalli, who's very kindly made the journey to join us today. So he's in the audience, Rajan. And we did this work, and also in PDD, and when we were looking at trying to build together a synthesis of published data in the area there. So a little introduction to levels of evidence. We have this, if you like, a pyramid of evidence, where at the bottom we have case reports and opinions. And towards the top here, we have the cornerstone of high quality clinical research, randomized controlled trials. And if we take multiple trials together, we can synthesize the data as part of meta-analysis and systematic reviews to generate data evidence that has more power. Here's an example of what some of those analyses look like. What we've got individual studies here that are then shown on this schema, this line. Anything to the left of this line points towards PDD being important, being effective at reducing rates of recurrence. And you can see individual studies sit on the line, so it's not particularly compelling. But when we combine those analyses down here, this is the combined data point, we can see it now just pulls away from that line. So the combined analysis tells us that we should be doing PDD treatments for our patients in preparation for their resections. However, and this is some of the work that Raj and others in my group did, we looked at the quality of the trials that went into the meta-analysis. And what we saw is that very important parameters that relate to RCTs, allocation, process of randomization, process around blinding, well, in nearly all the trials, there were very serious gaps in that data. So we now have to question the quality of that work. And having questioned the quality of that work, it was clear that we needed another trial. It's something that the NIHR had also appreciated, and they commissioned a HTA call, a brief for apply in open competition. And through open competition, we did apply, and we successfully got that bid, and we undertook the photo trial. It was in collaboration with ICR, with Emma Hall and her team, and also with CHART, the Aberdeen CTU, with John Norrie and his team. Special thanks to Rebecca Lewis, Themala Vadavilu, also Anne Duckton and Stephen Penwell, the trial managers, and also the statisticians. This was my first clinical trial, and John masterfully mentored me through this. He talked about the importance of trial design, and in particular the difference between efficacy studies and pragmatic studies. In these tightly controlled clinical trials, we can sometimes see a picture, a mechanism that holds true, but when we try to take those across into a real life setting where there's perhaps a more relaxed criteria, they don't always hold true. In fact, it's thought you can approximately half any effect size from an efficacy trial when you look in the real world setting. So in terms of a real world setting, which is what this was designed to be, we saw that when we look at this outcome graph here, looking at the rates of recurrence over time, this is being free of recurrence and accumulating recurrence as you come down, comparing PDD with white light showed no difference. You can see the curves did separate quite early on in keeping many of those RCTs, but the long-term data showed there were other biological factors at play here that were relevant. As clinicians, as scientists, we often rely on key opinion leaders or guidelines to tell us what is best. They undertake systematic reviews, but I'm going to urge everyone, that there's a lesson learned here, is still to question the evidence, question what evidence they've used to make those conclusions. And we might find that these systematic reviews, instead of being the absolute pinnacle of high quality research, because they've not looked at the right data or appreciated flaws, can unfortunately be only a poor copy. Okay, I'm going to take you through a very quick tour of some of the other clinical trials I'm involved with, and they're all designed with a very similar approach. These are pragmatic trials. These are all measuring a clinical effectiveness in a real world, real life setting in the RCT design. The first one I'll mention is ELLIPSE. This is a study I'm leading with Krishnan Hari from Cardiff, and we're looking at men with prostate cancer undergoing prostatectomy. We are asking the question, are lymph node dissections necessary for those that have high-risk disease? It's funny, because over the years we've stopped doing them. I remember when I first learned to do prostatectomy with Professor Suman Naim-Singh, who's in the audience today, we did lymph node dissections, and then we started tailoring that, and over time everyone stopped doing them, really, on the whole. And if you've got high-risk disease and you go and have the other main treatment, which is radiotherapy, they'll irradiate the lymph nodes. So there's a bit of a discordance, and there were some publications suggesting that you don't need to do lymph node dissection, but the devil's in the detail. They took all comers, they had low-risk disease, old school American cohorts with patients that we wouldn't even offer a prostatectomy to. So it's a really important study. We're looking to recruit over 1,000 men over the next 3 years to address that question. I'm delighted to say the study is booming. We're already on to our 100th patient, having just been open a matter of weeks. So well done to that team. Also want to give a shout out to the Urology Foundation, a charity in the UK supporting this kind of work, wants to make a difference for the urology community, and they have teamed up with CHART to help prime a number of these projects. These big projects need a long runway to develop the pilot data to make a compelling case to pull down these major HTA awards from the NIHR. We also have partial, which I'm co-leading with Naim Sumru, who's going to be talking at the end of today's session. This is a study looking at individuals that have kidney cancer. It's in those individuals that have got a normal kidney on the other side and one kidney affected with cancer. We know that with small tumours, we can cut those away and preserve the rest of the kidney. We also know with large tumours, you can take the whole kidney away, but the other kidney is good. It will do all the work and you'll be absolutely fine. What's starting to happen is people will start to take on bigger and bigger tumours in the kidney. However, that does come with potential risks and harms, higher rates of bleeding, higher rates of urine leak, potentially higher rates of having recurrence. And we wanted to understand those balances of harms and risks, and we're running this RCT. It's been a fascinating one because it's been really hard because of equipoise. Clinicians don't have equipoise. You either think you're a brilliant surgeon and you're going to do that partial nephrectomy, or you are much more conservative and say, no, I should just take the whole kidney. It's a really important study to deliver, and thankfully there's enough clinicians out there that want to do this, and we will be able to see this study through over the next couple of years now and give our community that answer. We also have this study as another HTA by NIHR, and this is a study looking at intravesical treatment for bladder cancer. We've been using BCG for years, and there's a fantastic opportunity to look at something that may be an alternative in this setting and theoretically might be better. It's a study where we're paired up with the ICR, and is co-led with Emma Hall and also Shen Tan, who was the brains behind this with his experience in the States, who are quite frequently using the experimental arm here. Shen's done brilliantly well and has now moved on to a post in Yale, has taken a role as professor there, but he's still very much active in leading this study. This last one, a special nod to this one here as well. This is one that's close to my heart because it's not always easy to deliver an RCT. That's what we want to do. They're the highest levels of evidence that we can generate, but it's not always doable for various logistical reasons. And we wanted here to look at follow-up of patients with prostate cancer. And we thought the best way we can deliver this is actually looking at a natural history experiment, looking at individuals that have gone through the pathway, and we can assign various types of follow-up and undertake an analysis of how they've done. And just want to point out the key players here as well. It's another collaboration with the Chart CTU, fronted here by Graham McLennan. I want to give a special shout to also Deanna, our absolutely fantastic trial manager, brilliant, and the magnificent Shona here at the back, a senior trial manager that makes that machine run. It's like clockwork, it's fantastic. There's a really exciting relationship here that's started with the London School of Hygiene and Tropical Medicine, with Linda Sharples, a world-leading authority on that kind of epidemiological methodology that we'll be pursuing. And with that, we've also got this super bright intelligence student that's joined us, Catherine Bunting, who's actually joined us in the audience today. Thank you, Catherine. So really excited about this work, and I think this might be a way that we look at developing a lot of studies in the future for those difficult to ask questions where we can't run RCTs. Right, okay, coming towards the end now, and I'll be circling back to where we started at the beginning, this idea about early detection of prostate cancer and that sad story with Chris Hoy. There is a large national study running, Transform, and I'm delighted to see a lot of the Transform group are here at the back, and it is the premier vehicle within the UK prostate cancer portfolio, expected to be funded up to and possibly more than 42 million, and we'll be looking to recruit over 300,000 participants for this. We're looking to, oh, it's died. Is time over? I don't know if we could bring the slides back. I'll give them a moment, I'll keep talking. So I think you saw there for a moment, yes, here we are, that we've got high profile press coverage. Of course, it seems intuitive. It seems that if you screen, you'll pick up cancer earlier. And if you pick up cancer earlier, you won't have the cases like Sir Chris Hoy. But it's not quite a done deal. It's not quite a done deal. So let's have a look at the evidence behind that and why we do need further research in this space. We have our best example. There's lots of studies out there that don't show a difference for screening, but we have our best example, this European study, the ERSPC, and it shows that you could reduce cancer mortality by 20%. But despite those findings, it may seem paradoxical that screening is not recommended. And the main reason it's not recommended is because you have to screen lots of people to find small numbers of cancers. A lot of people go through that, it's expensive. A lot of people then have side effects, concerns, anxieties around the diagnostic pathway, and we can end up diagnosing also clinically insignificant disease. Those are those cancers I was showing at the beginning, those that are on that indolent growth path that would never have caused harm, never would have presented, but you pick them up with screening. And the risk there is that then you treat these patients, and those patients have had no gains. They were never going to be hurt by prostate cancer, but we've hurt them by treating them because prostate cancer treatment with surgery, radiotherapy is morbid. So the harms outweigh the benefits. But there's been advances since some of those studies have come through. We're seeing some major changes in how we manage and diagnose prostate cancer. We've seen the emergence of MRI now becoming standard in terms of assessing patients at risk from prostate cancer. We have better ways of biopsying the cancers. We're getting better at making better treatment judgments for patients. So if you've got low-risk disease, we try not to diagnose that at all, but if we do, we know that we can watch, we can offer other approaches. In fact, if you have significant disease, we now have safer approaches, alternatives to the conventional approaches of radiotherapy and surgery, such as focal therapy, where we treat just the tumour and leave the rest of the prostate intact, avoiding serious complications. We're also seeing new biomarkers come through that will have a role in giving us a more accurate assessment of those at risk. So yes, game changers, we're reducing harms, we're seeing increased benefits, and we think the balance is in favour of prostate cancer screening, but we don't have the evidence, and that's what we're looking to do here with the Transform initiative. I'm delighted to be part of this super stellar cast with Hash Ahmed, who's been overseeing the leadership of our programme. Rianne Gavey did all the heavy lifting in terms of design and methodology. Mark Empton and Caroline Moore, along with Hash, who are the pioneers in developing the role of MRI for prostate cancer. And we've got Ross Eels from ICR, who has developed this polygenic risk score. It's a blood test, but perhaps you might have seen more recently also a saliva test, a spit test, which you can do at home, getting a lot of profile, and they may be a great way to diagnose prostate cancer. Yeah, a lot of other people involved, a really large team here, including Luke Vels in the audience. And I want to give a special shout out to Prabs Rajan as well from Barts, who is helping lead the Transform discovery, the translational part of this programme. Very quick overview of the project. We've got 4 arms we'll be comparing in the first pass. The standard pathway with PSA, elevation and MRI. We're dropping the PSA down to 1 with MRI. And then we've got very innovative arms where we're looking at MRI alone and we're looking at the polygenic risk score alone. In parallel, we're running this discovery program, and this is a really ambitious platform. We're looking to generate a national biodigital twin, looking to pull together biorepository of tissue, urine, saliva, blood, tissue biopsies, and marrying that up with digital information. And this is clinical metadata, digital scan data, digital pathology, to create this biodigital twin. And what this will do is work as a living biobank where we'll have tissue coming in, but we're looking to consume it, use it in real time, and get results out quickly. We're trying to make if you like, a biomarker testing machine that can work in real time, try to feed back early discoveries as quick as possible to make a change in the clinical space. We can simulate in these, once it's matured, we'll be able to simulate a number of HTAs at scale and pace and look at cost-effective analyses. Okay, so drawing to an end, and just an opportunity to thank a number of key individuals. Thank you, first of all, just to the clinical departments, both in Newcastle, the Freeman Hospital, and now Imperial at the Charing Cross Hospital. It does require a department that will nurture and support the academics to give the academic space and room to fly. And thank you all for that support. I want to make a big thank you to the patients. It takes so much time to go through these consent forms, take time out, take extra journeys. The generosities of patients is always overwhelming. I cannot be more grateful for individuals taking their time. Purely altruistic. Many patients will not gain anything from doing this other than that sense of doing the right thing, that well-being, and perhaps a chance to make a difference to your younger family, to your children. So patients, big thanks. I've had many mentors through the years, and I've tried to thank individuals as I've gone through each of the programs of work I'm doing. But 3 people I want to give special thanks to. Here's Rob Pickard, receiving the St. Peter's Medal from our National Society. Rob is no longer with us, but he was instrumental in picking me out from my basic science program and offering me a chance to lead a large clinical trial, and gave me the assistance and the platform to elevate me and give me a chance to become a star in that space. So big thanks to Rob. We've also got Luke Vale here. Big thank you to Luke. Luke has been involved with, I think, every single clinical trial I've been involved with, and he's been a source of sound wisdom and advice and steer throughout. And Naeem Sumaru, who has joined us today, and if you don't know of him, he's a surgeon extraordinaire, a robotic surgery pioneer, and someone for the Royal College of Surgeons leading their robotic and AI initiatives. And he's always prompted me to think bigger, think bolder, and to look forward, to look at what's coming and embrace those opportunities. Finally, I want to talk about the unsung heroes. So thank you to my parents who essentially illiterate immigrants as part of the kind of worker migration wave in the late 1960s. This was post-Britannia building back the UK, and they're part of that great post-war British success story. And they've not always understood what I'm up to, what I'm doing, and why I'm not around often, but they've always been immensely supportive. And you know, that has gone a long way to giving me the opportunities I needed to pursue this career. A special thanks here also to my wonderful wife Rachel, and you can see also my 2 boys here. Rachel's been with me all the way through that journey. So medical school, exams, MRCS, FRCS, coming through, applying for various position grants, failing, and that's the big thing. You know, there's a big journey of failure in this. So, Rach, couldn't have done all this without you. And I lastly thank my boys, who I give a tough time to, but I think, honestly, I'm so impressed with how well they're doing, and they're a constant source of inspiration for me to do better. So thank you.
It's wonderful, really, really wonderful. You would like to take some questions?
Yes,
is that
please,
okay?
yeah.
Can I just re-endorse really the thanks for the unsung hero? So I know how much it's been difficult to move from Newcastle here, and being in 2 sites, and the wife and the family really having the difficulty of you working in London and you are in Newcastle. Really, really very difficult. So from myself, I know how this would be difficult. So thank you very much. And the other one I forgot to thank is David Haruda, because sometimes it's very difficult to get academic appointments. It wouldn't happen without the understanding from the clinical colleagues and clinical environment and creating the job. So really thank you very much, David. I forgot to thank you in the beginning, but this is not unnoticed, and always remember to appoint 3 professors. That's a big thing. Questions? Yeah, please, on the back. Yeah.
I'm a prostate cancer patient. I survived for the past 9 years.
Yeah, absolutely. I think we are now starting to have unparalleled access into understanding the molecular makeup of prostate cancer. In the past, we've had to look at tissues, have very coarse metrics, and not really understand the complexity of what we handle in the laboratory space. We can now do this on single cell level. So we can really look at the complex dynamics that occur within the different components of a tissue. What that will do, I think, will give us opportunities to understand new approaches to control prostate cancer, new ways that we can possibly even completely cure individuals of prostate cancer. I very much am optimistic that the days of hormone, which have stood the test of time and have been really powerful in prolonging lives and saving lives as well, but I think there will be new treatments in time, yes.
A statement and a question. One on screening. I'm a public health doctor, there are many screening programs that reduce cancer-specific mortality, but don't change all-cause mortality. They redistribute the death certificates and what's written on them, but not the number of death certificates doesn't change. Some of the bowel cancer screening in America, for example. The question was, with all your genetic studies, the very different rates of prostate cancer between black Caribbean men in particular and white men. Is there anything coming out in those genetic studies for any genetic component? Some of it must be environmental because it's different in the Caribbean and here. But
the insight, I mean, that's a really good question, and the insights that we're learning about different ethnicities is really quite remarkable. We're starting to understand that if we look across the spectrum, there are a number of potential different drivers. There's many ways to get to the end product of having cancer. The makeup, depending on which combination of mutations that you have, can be powerful in determining the nature of that cancer. Is it going to be one of these aggressive cancers that are life-threatening? Is it going to be a slowly growing cancer? We slowly start to add to that encyclopedia of mutations that will help us stratify those that would be at higher risk and those that won't be. We do need more data, but the international global effort in this space is huge. I think we're not far away from being talking about those kind of outcomes based on those genetic information soon. In black men who are more vulnerable to get prostate cancer and more likely to get aggressive prostate cancer, the picture is mind-blowing. You know, from a UK perspective, we have them categorized as black men, just black men. But the history of black men is wide, all across Africa, and the genetic richness, diversity within black men from their original ancestry in Africa, from even small regions, is really high, perhaps related to tribal ancestry. And we're finding in that picture that the variation of mutations are colossal. In fact, they have the highest variations of mutation rates and polymorphisms out of all of the measured communities across the world so far. It just lets you into an insight into how challenging that's going to be. We can't redo that work here because we don't have enough black men here. So what we are seeing is more innovative approaches where charities, funding organisations are reaching out into Africa and supporting universities in Africa to undertake that kind of work, where they've got a larger pool of people that could support a wider review of the genomics for those individuals.
Question back.
Since prostate cancer is hereditary, you've mentioned the high risk in terms of black African and Caribbean men. Should, in terms of family history, those people be given a priority in terms of having PSA blood tests, possibly earlier age, 45, 50, or any race where it's actually prevalent within the family? Because obviously there's a defective BRCA gene in the family, isn't it? And it's very similar to women who might have breast cancer.
Yeah, it's a really important question, and it's an area that's quite sensitive at the moment in that we do want to reach out to black men. We know black men are at higher risk, and I completely agree with you. That's what we want to do. The design of Transform to understand that biology and those risks talks to how important black men are. We are purposively sampling, so we're going out and oversampling black men so we can have an interpretation of that biology. Right now, I think we've got enough information to say we should be looking to screen earlier for those that have black ancestry, family history, drop the threshold, which is, I think, what you're alluding to. But there's other challenges. And again, part of Transform, we're looking at how we can resolve those challenges. We have issues where we're struggling with the current framework. The UK struggles to reach some of those black communities. And we need to work better with black communities. Black communities need to be telling us how we can reach better. Because they're not hard to reach, we just don't know how to reach them. So hard to reach communities is not the right way of putting it. It's something I learned very recently. I had my wrist slapped as I talked about it, and someone said, no, they're not hard to reach, you just haven't worked out how to reach them. So that's actually a really important part of the early phase of Transform, is learning how we reach those individuals.
I was taught in school that genes and the environment interact. I find, having been sacked by the Cancer Research UK for trying to go along the environment age, that we're still not looking at what is the interaction. And particularly, it's now 18 years since it was first pointed out that men who had bad acne had a risk factor of about 1.7 of cancer at 30 years later. And then they found the bacterium in the prostates in the subset. And we now know that this particular bacterium switches off the normal BRCA repair gene. And these sorts of situations build up the number of infectious agents that have been in and out of prostate epidemiology. And it's in cervix cancer, we have many of them similarities. And we don't take any notice of these relatively low-grade irritants that over 30 years could be making the repeated changes that ultimately produce cancer. Now, from the point of view of doing surgery, particularly if you're doing surgery in the most advanced ones that you can do surgery now, we've got such an increasing number of immunotherapies that could make preoperative immunotherapy make you vastly improve this subgroup. Potentially, now you've got the robots, you have the ability to get them out safely.
I think I'm going to look at maybe two dimensions of what you shared with us, and they're both going to revolve around Transform. One is the epidemiological risk factors that pertain to getting prostate cancer, lifestyle, environmental factors. And we're looking to run a program not far away from what's been happening with Our Future Health, looking at taking detailed histories, trying to define particular demographics, particular exposures that might be relevant. Ultimately, we're also designing our collection to be able to link to those kinds of work so we can pull in the extra layers of data available that might shed light onto some of the causative agents of prostate cancer. Regarding the biology and looking at doing deep dives of all the factorial elements that could be relevant in transformation, yes, Transform Discovery, we'll be using multimodal approaches. And for the first time, I think we've got the computing power to bring lots of different analyses together to understand how that interplay works. Thank you.
Shall we see the last question? Is that okay? So we can, thank you.
Transform sounds very impressive and transformative, but I can't believe that we in this country are the only people in the world that are trying to, deal at the cutting edge of understanding prostate cancer. What else is happening? Europe, US, Japan?
Yeah, it's a good question. Often what they do, because they're colossally expensive programs, the world tends to look what's happening in the UK, in the US. If something happens in the UK and it's shown to be positive, we're world leaders and a lot of people will follow quite simply. We did have the CAP study, Freddie Hamdi study, looking at screening with one-shot PSA. That's matured, published, and looking at long-term outcomes. That was at the heart of informing the National Screening Committee that there are risks, there are harms. It didn't show a difference. It's against screening. What is fascinating now is that over time they are starting to see some signal come through. It takes that long to see a signal, and it looks like it's a bit more encouraging. The American studies, they've not been able to run that because people want to go and get their PSA checked. So there's not a true control. And that's the risk that we possibly fear in Transform. There's a dilution occurring where people in the back door are getting tests. We're watching that carefully, but we're one of the few healthcare systems, because we're in a healthcare system, not private, that means that we have a more organised way of delivering care and allows us to generate better controls with our experimental arms. So, no, I hear what you're saying. There is work elsewhere, but the world's looking to us.
Thank you. So before I conclude, ask Professor Deborah Ashby, thank you for coming. Any comments or anything you would like to add before I hand over?
Just to say how much I enjoyed that. I met Rakesh when I interviewed him. I was chairing at the time. I think I came off fairly soon after, but an absolute delight. And working with surgeons and statisticians is a magic combination.
Thank you, Deborah. So now may I hand to Professor Naeem Simru to conclude.
Thank you very much. Yes, I think I won't take very long, so I have to just give a note of thanks to Rakesh. Rakesh and we have known each other for over 20 years when he was young, bright, surgical trainee, an academic trainee. I think Rakesh, probably many people in Imperial would not know, come from a pedigree of illustrious academic surgeons from Newcastle University. At one time, most of the professors in the UK in urology, starting from Glasgow, Aberdeen, Sheffield, Leicester, Cambridge, Oxford, UCL, Bristol, they all started their academic careers from urology, and Rakesh is one of those illustrious. So he is in a very high esteem as an academic surgeon. Actually, Professor Marcus Drake, who is sitting over here, actually he was trained in Newcastle as well. So I'm coming from Newcastle, so I'm seeing the sense of pride that Rakesh is leading that tradition. I think it's quite hard enough to be a basic scientist, and you can spend a whole life in trying to do that. But Rakesh is not only an excellent basic scientist and trying to understand the biology of a disease, he's also have a huge number of clinical trials which he's involved in. One of them, I'm involved with him as well. But he's also a fantastic surgeon. He was very keen that he should train in robotic-assisted surgery, which is a skill in itself, and it's quite a hard work. So he wanted to be as good in that aspect as well, and he's an excellent surgeon. So I think that encompasses the totality of not only being a basic scientist, clinical trial surgeon, and also a fantastic surgeon as well. And I think, and that speaks to his character. And I think it's been a privilege for me to have known him as a colleague, as a friend, and also to see the opportunity which lies ahead of plans that he has. And Rakesh was last year, many people don't know, was recognized by the Department of Health that he got the highest award a consultant can get, So he got N3 award. It just means that you are the top award over here. And he was one of the few surgeons, or the few consultants in UK who got that award. So I think he is very well recognized for his work in academic science, but also in delivery of research as well. So it's fantastic, and it was so fascinating to see what you have done, and also the opportunity that you will have in future. Wonderful. Thank you very much. Thank you.
Wonderful. I think that brings us to a close. Thank you so much. It's been an absolute pleasure seeing so many people turn up. Thank you, and I hope you can join us for the drinks reception.